Voltage gate ion channels 2. Structural function of channels for ionic selectivity. Today we're going to discuss some of the mechanisms of ionic selectivity in potassium channels with a brief discussion for calcium channels as well. The idea of ion channel selective pores in the cell membrane is very old, but is now understood at atomic level. So what is a channel? What does an ion conduct pore looks like? Here I will use some of the recent structures of potassium channels to briefly discuss these questions. Shown here is an early cartoon of a voltage gate channel where the channel is drawn as a transparent protein with functional regions such as the gate, a voltage sensor, and an aqueous pore. A narrow part of the aqueous pore would serve as a selectivity filter, which is a domain in the pore that would allow the permeation of certain ions and yet exclude other ions. So the central question here has been how potassium channels are able to achieve very high selectivity for potassium and yet exclude other ions such as sodium. As you can see in this slide, potassium have ionic radii of about 1.3 angstroms, while sodium have ionic radii of about 0.59 angstroms. So they're very similar in size, and yet sodium ions are even smaller than potassium ions. So a selectivity filter that excludes the ions purely based on size would not be able to work for potassium channels. Although potassium channels are extremely selective in which ions they allow to pass, they allow transport rates close to the aqueous diffusion limit. About 10^7 to 10^8 potassium ions go through each channel per second. Selectivity and speed are both crucial for the biological function of these channels. High flow rates are essential for producing these voltage changes very rapidly. A typical action potential in our mammalian neuron requires millions of ions to flow in a millisecond. To accomplish this without using millions of proteins requires that each one have a very high flux rate while maintaining very high selectivity. What we have to remember is that the ions in solution are surrounded by water molecules. In this table, there is a list of the solvation energy and the radii for the various ions in solution. As what you can see is that solvation energy is very high for these ions in solution, with sodium and potassium about 100 kilocalories per mole. So it takes a lot of energy to remove these water surround these ions in solution. So what would be the behavior of these ions when they go through these ion channels? Here is one model about 1970s that is showing potassium ions in red surrounded by water molecules on the intracellular side of the cell. And what's shown here is the gate of this channel being closed on the left, and when this gate opens, potassium ions still surrounded by water molecules can enter through this channel in this very large vestibule and then enter finally the selectivity filter of this channel, and then the potassium exits on the other side where it regains some of these water molecules. Also shown in this figure, this strange molecule called C9+, which is actually a derivative of a triethyl ammonium ion. It's a relatively large ion that's also relatively hydrophobic, because what's shown in that time is that these very large relatively hydrophobic ions can go inside of these channels and get trapped inside of these channels. So the idea here is what these channels would have this very large vestibule that would be able to accommodate relatively large ions such as these triethyl ammonium ions. So the question here is in regard to this selectivity filter here. How this selectivity filter is able to discriminate potassium over other ions such as sodium. So how the selectivity filter would work? We can think the selectivity filter as a structural motif that will emulate hydration shells for potassium ions. In this model, the carbonyl oxygens in the bottom here from the amino acids line the selectivity filter emulate the water oxygens in solution, allowing almost no loss of free energy when potassium ions traverse the selectivity filter. This model also explains how this kind of filter would discriminate potassium over sodium ions. A sodium ions with a 0.95 crystal radius in the filter has higher energy than one in the water because it binds effectively to only 2 of the 4 carbonyl oxygens shown. The other 2 other carbonyls are prevented from collapsing around the sodium ions by structural constraint in the filter wall. In other words, we can say there is a high energy barrier for sodium entering and exiting the selectivity filter, while there is almost no energy cost for potassium ions to do so. While these models were advanced using clever electrophysiological and biochemical experiments, there was no proof of its veracity until the advent of molecular cloning of potassium channels. Shown in this figure is the amino acid alignment of diverse types of potassium channels in the putative selectivity filter domain of voltage-gated potassium channels, Shaker KV channels, a calcium activated potassium channel, SK2 channel, and track task channels, which are 2-pore domain leaky potassium channels. What is apparent here in this figure is the high conservation of this TVGYG motif here. This motif has been called the signature sequence of potassium channel, as in almost all cases, genes that encode such sequences turn out to be potassium channels. Could this TVGYG motif be essential for potassium selectivity of potassium channels? If that's the case, then how to test this idea? One possibility is to compare the sequence with channels that are not potassium selected, such as the CNG or cyclonucleotide gate ion channels. These channels permeate both sodium and potassium with almost equal efficiency, and the alignment in the pore region shows that the YG amino acid in CNG channels are missing. So investigators deleted this YG sequence here in the Shaker potassium channel, and they expressed these mutant channels in cell lines, and what they found was that these channels became non-selective to potassium ions, just like the CNG channels. Notice the presence of these inward currents in the mutant Shaker potassium channel that are due to the influx of sodium ions. So from this experiment, we could conclude that the GY residues in the signature sequence underlies the potassium selectivity in potassium channels. What are some potential alternative conclusions? Instead of using Shaker potassium channels, could you use the CNG channels to either reinforce or refute the above conclusions?